plant's cellular structures and their characteristics reveal so much pertinent information that will help us understand its anatomy and biology all the more. A classic and inexpensive way to do this is through freehand sectioning, which will be shown later. With freehand sectioning, different parts of a plant are sliced or sectioned, stained, and mounted on slides for viewing under the microscope. Different plant tissues will then react at different rates with the various dyes used. Here, practice makes perfect, and that means having 1 cell thick sections. Before we jump into the procedure, we must first categorize our plant part of choice as flat or round. This will help us determine the technique we will use in sectioning them. If your plant part of choice is round, you may section them in the 3 basic planes to study their 3-dimensional shape. Transverse section or cross section. This is made by first identifying the plant's longitudinal axis and cutting at right angles to it. Radial longitudinal section. This is made by cutting parallel to the longitudinal axis, and finally, tangential longitudinal section. This is done by cutting at an angle to the longitudinal axis. There are also 3 basic planes in sectioning flat plant parts, and these are transverse or cross section. This is also done at a right angle to the longitudinal axis. Sagittal or longitudinal section. This is made by cutting parallel to the longitudinal axis. Paradermal section. This is made by cutting parallel to the epidermis. Now that the different planes and your options for cutting is clear, let's start sectioning. First, cut the specimens into small pieces. You'd like to have 3 or 5 centimeters of length for your plant specimen part. Do soak the specimen for sectioning in water to prevent them from drying out. Next, what you would like to do is to get a stable support for your specimen. Do this by taking a radish or potato and cut them longitudinally in half. Then on the inner surface of one or both halves, make a groove deep enough for your specimen to fit in, and then close the specimen to be sectioned here. Third, hold the support clamp containing the specimen in your less dominant hand, between your thumb and forefinger, and in the other, hold a sharp razor blade. As you can see, the end to be cut is positioned a little forward than your thumb and forefinger. Fourth, cut by quickly drawing the blade through the clamp with the specimen. It is important that you do not saw, but slide the blade smoothly and quickly through the material. Thin sections will then stick to the blade and accumulate there. After cutting, dip the blade in a watch glass with water to remove the sections from the blade and, most importantly, prevent dehydration. When you have your choice cuts, we can start mounting. Step 1. Fix your sections using FAA. Then we now intentionally dehydrate the sections by placing the specimens in 40% ethanol for 30 to 60 seconds. Rinse them with 50% ethanol for the same amount of time and then transfer to a dish or vial with the stain. This contains 1% safranin in 50% alcohol and hematoxylin. The sections are then left to stain for 24 hours or less for some specimens. Then, transfer the sections in 50% acidified alcohol for decolorization. This may sound a little counterintuitive, but too much stain will obscure the characteristics of your specimen's cellular structures. Step 6. Put the specimens in 95% alcohol for 5 minutes to stop the decolorizing effect of the acidified alcohol. Step 7. Soak them in fast green for 30 to 60 seconds and then wash in 95% alcohol. After washing, transfer them in a 50-50 mixture of absolute alcohol and xylene. It is advisable to do this under the fume hood because xylene fumes should not be inhaled. Then after placing them in this mixture, transfer the sections in xylene and let them stay for 10 minutes. After 10 minutes, take the specimens and place them on separate slides. Place a drop of xylene on each to prevent their further dehydration. View the sections and select which will be most suitable for mounting. Finally, mount your finalists in Canada Balsam. Place a cover slip on top, let it dry, and then finally, label your slide. Editing Ethel here. Freehand sectioning as a phrase. It's a bit odd. It's as if we're supposed to section our free hand. Just a thought.